I've been able to do it. So this is the complex at the cell surface of T cells that recognize the peptide when it's being presented by the MHC molecule. It's called the TCR complex. And the TCR complex has three major components. The TCR itself, which is the structure that will recognize the peptide, the microbial peptide or viral peptide or bacterial peptide presented by the MHC class 1. It has a variable region and a constant region. And we'll talk about having the importance of having a variable region. It has two similar chain, alpha and beta chain. Okay, that's the first component. That's very important. The second component is the CD3 complex. And CD3 complex is very important. Why? Because once the TCR engage with the right peptide, the cells need to be activated. And CD3 and this delta chain are important in activating the biochemical signal that will activate T cells. Okay, this is the two part. The second is the accessory molecule. So T cells can be either CD4 or CD8. They can't be CD4 and CD8. Therefore, you will have this complex with either CD4 at the cell surface, and CD4 is important because it's an accessory molecule that recognizes MHC class 2. That's why CD4 positive T cells can only be activated when the MHC class 2 protein are presenting the peptide. Similarly, T cells can be CD8 positive and can recognize only peptide presented by MHC class 1 because the CD8 molecule only recognizes the MHC class 1. So you see how specific it is, okay? So the question is, how is the T cell receptor able to recognize such an array of different microbes? I mean, you would need a lot of gene to do that in order to accommodate different variable region which bind your peptides. So how is the host doing it? And this is another wonder of the immune system. I can't go detail on that. I'm just going to summarize. This is a mechanism called combinatorial diversity. And basically what it is that the T cells have different gene segments coding for the variable region, okay? And these gene segments are assembled in a very random manner to create the final gene that will produce the variable and the constant region. So it's like having a Legos. I give 10 Legos, different color to different people. They will assemble them differently. And this is exactly the same thing. You would do it in a very random process. And because of this random process, you create, look at this amazing possibility of T cell receptor. You have 10 to the 16 different possibility of TCR. Meaning that there shouldn't be any single peptide that would not be recognized by TCR. And of course, you would ask, how come we have a poor response to certain antigen? The limiting factor in all of this is the capacity of the MHC molecule to present the peptide. And that's what the host cannot control. We have all the means to recognize all the microbes. The only thing we can't control is our MHC molecule that we inherited. This MHC molecule will only present certain peptide and not all peptide. And therefore, the limiting factor in all of this, in activating T cells, is having the right MHC molecule, presenting the right peptide. Okay, so as I said, this T cell receptor, which recognizes the antigen or the peptide, when I say antigen and peptide, the same thing, they are displayed by MHC molecule. Nothing else, the T cell receptor can recognize the microbe, otherwise it's not going to work. So you have MHC molecule, and I will show you how, TCR, okay? And finally, because if the T cells has a CD4, it's only going to recognize MHC class 2 molecule, presenting the microbe. If the T cell receptor complex has CD8, it's going to be CD8 cells, will only be able to recognize microbe presented by MHC class 1. So you see the selectivity of this T cell antigen presenting cells interaction. And I will summarize that here. So here is an APC, antigen presenting cells. This is a naive T cells. And we assume that antigen presenting cells has captured the antibody, process it in such a way that will be cleaved in small peptide and presented by MHC class 1 or 2. And this is the MHC molecule. This is the antigen presenting cells. I'm just presenting summary. And this is the peptide which is here. This is the a variable region of the TCR receptor. This is the CD3 complex that will give the signal. And this is the CD4, CD8. So if this protein is MHC class 1, this is going to be CD8. If this is MHC class 2, this is going to be CD4. You see how specific it is? So there's no way you can have the wrong activation, meaning that if you have a bacterial peptide, you're not going to activate a T-cell response that's not going to be appropriate. Remember, I like to ask this, I'm going to tell you, help you a bit here, is whenever the student has been examined on question, I tell you where the question is. This is a question that I usually like to ask about co-stimulation. So this is the first signal. T-cells won't be activated only by recognizing the microbe presented by MHC molecule. It needs another signal. And this other signal is provided by this B7 molecule that is upregulated on antigen-presenting cells, okay, during maturation, and that will activate a protein called CD28 on T cells. Once you have these two signals, this is what we define as co-stimulatory signal. The T cells can be activated. If you don't have these, you're not going to have T cells activated. So it's very important for you to understand that you need these two signals, okay? MHC molecule peptide TCR, B7 CD28. Very simple. In addition to that, and you will see when regarding the function of T cells, the APC will secrete different cytokine, telling the T cells, the naive T cells, what they should be doing or which cells they should be becoming. Because the antigen-presenting cells are there to organize the response, and they are there to tell the right information. And they're going to tell the T cells, this is the type of microbe I have. This is the type of T-cell response I want you to do. And I will show you how it's been done later on. In any case, once you have these two signals being met, co-stimulatory signal, important for question, you're going to have either CD8, CD4 positive T cells becoming T helper cells. Remember, CD4 always become T helper cells. These are the cells that are being targeted by the HIV virus. And it will tell you why is this important. CD8 naive T cells will become CD8 effector cells or cytotoxic T cells. These are the cells that will go and look for all the viral infected cells to kill them, to destroy them. Okay? I hope this is clear on this stage. If not, then, so let's go back now. Let's see what signal APCs give to T cells and what is the outcome. And this is usually what happens. So you have the APC, they have captured the microbe, they went to the closest lymph node, met with the naive CD4 positive T cells, because we need CD4 positive T cells to, even if it's against a bacterial or viral infection. And what they're going to do is, if antigen-presenting cells secrete a cytokine called interleukin-12, this naive T cell will become Th1. If antigen-presenting cells secrete IL-4, it will become a Th2 response. If antigen-presenting cells secrete IL-1 and IL-6, they're going to become Th17. And the same for IL-10 and TGF or Treg. So you can see that antigen-presenting cells can tell the naive T cell to become anything, depending on the nature of the threat. If this is an intracellular microbe, the CD4 positive T cell will become Th1. These are CD4, Th1. Why we call them Th1? Because they are the best to handle microbe that are that replicate inside the cells, as well as extracellular pathogen. But I just want you to remind just about intracellular to be easy. So Th1 is a CD4 positive T cells that will help CD8 become cytotoxic T cells. You understand? Help macrophage to be recruited at the site of infection and activate them, and tell B cells to produce specific antibody called IgG or IgA. IgA at the mucosal surfaces, IgE inside the cells, inside the host, in tissue. Okay? Very simple. Let's assume now that the antigen-presenting cells have sensed a different kind of microbe. Let's say it's a parasite or a worm. Then this will be inefficient. Why? Because you need a Th2 response. Why do you need a Th2? Because the B cell will produce the best antibody response against this big parasite, which is an IgE. IgA don't do anything against a parasite overall. IgE is the best response against that. Th2 will also activate recruit eosinophil, which with the help of IgE will kill the microbe. And more importantly, and you will see one side effect of this Th2 response is allergy. I'll talk about it now because I don't want to make things complicated. But just bear in mind that when the host make a Th2 response against, let's say, peanut, you're going to end up with allergy, which normally you shouldn't make. You should have a tolerance against all the food protein we eat. But in some situation, some patient will develop an abnormal Th2 response against peanut and will end up with some serious cases of anaphylaxis shock. If this is, again, microbes as well, then the naive T cells by the APC can become Th17. This is a new type of T helper cells that produced interleukin-17, has been shown to be instrumental in recruiting and activation of a neutrophil. So this will help against any other microbe. And finally, Treg, we don't talk a lot about, I don't want to remember, but this is very important cells that act as the policeman, the T helper cells that act as a policeman. And this is what it does is ensure that we don't react against self-antigen, and we also suppress the immune response when it has to be stopped. So you can see from naive T cells and from the interaction with APC that you can drive different type of T helper cells depending on the type of microbe you have. Okay, so and that's actually just amazing, the plasticity of these naive T cells to become different type of helper T cells. So let's see now, and we've seen that. Let's see now we have a virus, and the virus is different. So now here's the issue, and this is going to explain something very important. So the best weapon against virus are cytotoxic T lymphocytes. And what they do is antigen-presenting cells sensing the virus, capturing the virus, presenting the virus via MHC class II. And I said the naive, the antigen-presenting cells are the only cells that can process intracellular microbe by the MHC class II. All the other cells, they can't. They can only present by MHC class I. And they will tell you why this is important. So the naive T cells gets activated by antigen-presenting cells, by the receptor, to become effector CD8 positive cells that become cytotoxic T lymphocytes, or the killer cells. But this activation here is impossible if you don't have T helper cells, Th1, because the Th1 will help in the maturation of the effector CD8 positive T cells becoming cytotoxic T lymphocyte. Okay, this is extremely important. So if you don't have these CD4 positive T cells, you're not going to have fully activation of cytotoxic T lymphocyte. This is extremely important at this stage. And I will show you here the example why. So remember, HIV virus affect CD4 positive T cells and kill all CD4 positive T cells. Doesn't touch these CD8 positive T cells. And you can see here in the course of the infection, you have a latency period and you have this symptomatic phase of virus. You can see that you have a number of anti-HIV virus, but you can see that in the course of the asymptomatic phases, the number of CD4 positive cells gradually decrease, okay? And while this CD4 positive T cell decreases, the viral virus load increased. So it's actually the best proof that you need CD4 positive T cells as a form of Th1 response to produce antibodies, but also to clear the infection to produce the cytotoxic T lymphocyte. So it's very, very important for you to understand that the CD4 is essential in not only producing antibody, the right antibody, but also producing cytotoxic T lymphocyte. So let's see how actually the virus is sensing and killing the viral infected cells. Remember here, once they activated, they will leave the lymph node and go to all the tissue and sense, look for the viral infected cell. And this is how they do it. It's not, there's no sound, I'm going to comment on it. and stop at the time. So